Firstly, I'd just like to acknowledge GRDC for funding this work. It is a national project. We've got people in all the canola growing states around Australia involved. So to get the ball rolling, I thought we'd just go through and say, well, what is blackleg, what does it do? How much disease or yield loss does it actually cause? So this, when we talk about severe levels of blackleg infection, this is the sort of disease levels that we see if you get things wrong. You've got the wrong crop in the wrong situation in the wrong region, and you get lots of plant death. You obviously get some survivors, but when you cut them open, you'll find there's lots of partial cankers forming, etc., and you obviously get quite significant yield loss. This is a situation, this was in an MBT trial, where when things go really wrong, blackleg can basically wipe out the crops, as it's done a number of times since we've had it in Australia. However, for most guys, when we get things right, there's no reason why we can't grow canola crops like this and really don't have any yield loss from blackleg at all. So we bring all our management packages together. There's most guys, achieve this type of control, and there's no reason why we can't achieve that on the majority of our crop. So to start the ball rolling, why is blackleg such a big issue? Why has it been around with us for 30 years and the breeders have just really coping with it, trying to keep up with it? Why don't we ever really get on top and nail this problem? This is the blackleg life cycle, and if we just concentrate here on the top row, this is the main reason why we have such an issue with blackleg. Firstly, blackleg survives on the canola stubble, and you can see there the little fruiting bodies forming on it. So it doesn't matter how much disease you had last year, what you did, basically over the following summer, the stubble will be totally colonised by the disease. And then all of that stubble will be loaded with these little fruiting bodies, which are throwing out all the blackleg spores, which are wind blown onto your next crop. Now the thing with these fruiting bodies and the spores is that they're a result of sexual reproduction. So they're genetically incredibly diverse, and in fact every spore is genetically different to every other one, and there's trillions and trillions and trillions of these things. So the result of this is that when you grow a cultivar, even if it's never been grown before, even if it's totally resistant from the seed company, there's already individual spores out there which can attack it. And every time you grow that cultivar, all you're doing is selecting for those spores which can attack it, and then the next year, the stubble will actually be throwing out all those virulent spores. So what we see is that when you grow a good resistant cultivar, you get 3 or 4 goes at it, and then the fungus will start overcoming that resistance. And that happens with all cultivars in most situations. So if you consider this, compared to a lot of the other diseases you might be managing, if you consider it to, rust in your wheat, rust needs a green bridge. So like after a summer like this, there's virtually no green bridge, and the rust has to build up its inoculum the following year. With our blackleg, we grew 2 million hectares of canola last year. We've now got 2 million hectares of stubble totally loaded with spores in our growing region ready to go. So we've got absolute maximum levels of inoculum every single year before you even sow your crop. And the other thing is, like I said, these guys are sexually reproducing. If you look at your rust, it's an asexual disease, so it's not going to overcome resistance quite as quickly. The other thing with blackleg is it's really affected by which region you're in. So it's really an effect of the intensity of the canola production. So this is a couple of years ago now, before Eyre Peninsula really got into canola. But when you have this level of production, basically you can't avoid the canola stubble. So if next year you grow your canola in here, you've got stubble either side of it, you're getting total really high levels of inoculum. And this really level of disease is dependent on the region you're growing in, which obviously you can't change the location of your farm. On the flip side of that, this is a crop a couple of years ago after the drought in Victoria, where no one had grown canola for a few years, hadn't had the opportunity, and grew magnificent crops with virtually no disease at all. So the blackleg intensity and infection that you're seeing is really a result of that level of canola intensity. Okay, so just in summarising why blackleg is such a severe disease and why we always talk about managing it and trying to stay up with blackleg rather than really trying to control it completely, is that it's sexually reproducing, it's incredibly diverse, and it changes its populations very, very quickly. We use molecular tools now, we can see populations changing by their virulence, by the individual isolates by 30 or 40% from one year to the next. It can change really quick. It survives on the stubbles, there's a massive amount of inoculum, and you're already going to be in a situation that's controlled by the intensity in your region, which you can't control. Okay, so we'll move on to the management of the disease. So this is the Australian Blackleg Management Guide, which we put out in August last year and will now be updated in March every year. So this really is the one-stop shop for blackleg management. We've tried to put in there everything you need to know for blackleg management. So as an advisor, if you download this off the GRDC or NBT websites each year, we'll keep it updated and it should have everything you need. It's got the latest cultural practices, it's obviously got the latest blackleg ratings, which get updated every year, and it's also got the latest resistance groups. Okay, so moving on to management of blackleg. One of the things we're really trying to do, and we'll put a big effort into this over the next 12 months, is really trying to get people to monitor how much blackleg they've got in their own crops so that you can make informed decisions. We talk a lot about what happens in Eyre Peninsula, but the reality is most growers aren't in that level of intensive canola production and don't have to worry so much about blackleg. So we really want people to know, have I got a disease problem or not? If I have got a disease problem, then I can go through all the cultural and whatever practices. If I haven't got a disease, I can keep doing what I'm doing. So really, the monitoring shouldn't be too difficult, shouldn't take too long. You do it just after or prior to windrowing. We always used to say it straight after because it's easy to get around the crops, but then a lot of farmers have said they're busy then and it's actually better for them just to try and struggle through the crop when they haven't got too much on. So it's sort of really any time after flowering is fine to get an idea. We just want you to walk a bit of a transect, pull plants out of the ground, cut them open, look to see how much stem canker they've got, and keep a record so that you know whether your disease levels are increasing or not. And these notes are actually in your books as well. So there's a bit of a guide. Here's a crop where obviously it's very visual. You're seeing dead and dying plants and you pull them out and you'll see they're cankered. And that's obviously a very strong indication that you need to change your management because you're getting major yield loss. But this is what we want you to do with your healthy looking plants, to still pull them out of the ground, cut them off right there, that's where the canker forms with your parasecateurs, and then have a look inside and see what's going on. So these plants have obviously got lower levels of infection, and if your crop is generally looking like that, we recommend everything's good, keep doing what you're doing. If you're starting to see these levels of infection, well then you're starting to get yield loss. So as a rule of thumb, when you see more than 50% discoloration, that's an indication that yield loss is occurring and you want to start changing your management practices to get back to this type of infection. And you can see here these plants are actually quite intact, so you can't just walk around the paddock and just look. You've got to go to the effort of picking them out and cutting them off. And the other thing is, when we talk later in the talk about cultivars losing resistance and changing resistance groups or whatever, it's really crucial to know that did my cultivar start out looking like this and does it look like this now? That's an indication that you're losing resistance and you want to change cultivars before it's already overcome. But if you don't have this data, it's very hard to make those informed decisions. So our first blackleg management control is the cultural practice of basically avoiding stubble. If you're in a high risk situation, like in Eyre Peninsula, it's very difficult, you can't get away from that stubble. But for most people in Australia, they can still get some separation from this year's crop and last year's canola stubble. So this is basically blackleg infection compared to distance to stubble, and you can see at about the 500 metre mark we stop getting that additional infection level. So after that, I think you've got basically like a cloud of spores right across the entire region, and there's not much you can do to avoid that. But as you come in closer to last year's canola stubble, you get more and more infection. So anything you can do to stay away from last year's canola stubble is a benefit. Older stubble releases a few spores, but generally it doesn't have a huge effect on the amount of disease you get. So there's just some visual photos we took. So in this experiment, we grew plants and then put them at different distances from stubble, and we used plants which had no blackleg resistance at all, and you can see there the canola on canola is totally dead. But even at just 250 metres away, the plants are starting to survive. So any distance you can get away from canola stubble, the better. And this is why we always say you can never grow canola on canola, because that's the disease pressure you're going from there to there by just having that small break. The next control mechanism, and probably by far the most effective, is still the resistance ratings. So we put out the new resistance ratings each year. This is the 2012 disease nursery near Horsham, And you can see basically all we do is we sow each cultivar in a row, and then at the end of the year we assess whether it looks like this and it's got very good resistance, or it looks like that and it hasn't. So these are commercial cultivars, these aren't experimental lines or whatever. So if you're in a high risk situation and you grow the wrong cultivar, that's what's going to happen to you. So it's really crucial to know the resistance rating. The other thing with these trials is we also have them with plus or minus jockey now. So you'll get in the disease ratings, you'll see you'll get a rating with the addition of jockey or without jockey as well. Now, as we talk more about resistance in our cultivars, we're learning more and more about it, and you'll hear these terms of seedling resistance or lesion resistance and adult resistance, canker resistance. So I'll just spend a couple of minutes going through so you understand the concepts. So this is our seedling resistance. And the way seedling resistance works is pretty simple. The spore lands on the plant. The plant has a gene which enables it to either recognise that it's being attacked or not. If it recognises it, it responds and you get no infection. If it doesn't recognise that it's being attacked, you get this type of infection. You get your leaf lesions forming. So in this situation, the plant is not recognising it's being attacked. You get a necrotic lesion with more fruiting bodies in it. The pathogen then grows down into the vascular tissue, down to the crown, and causes the adult canker. And this is the adult plant resistance. So this is basically the way it works. When we infect cotyledon, there's no resistance there. The plant is colonised, it grows down and causes stem canker. In this situation here, we've still infected the cotyledon. The plant, it's got the corresponding resistance gene. The plant recognises that it's being attacked. It doesn't let the pathogen in, and therefore no stem canker. And there are Australian cultivars with that type of resistance, effective resistance. In fact, nearly all of our cultivars do have seedling resistance, although most of them, that resistance has been overcome, so it's no longer effective. So this is the situation we normally see in Australia, that the seedlings, we infect them, the seedlings do get colonised, but then it doesn't go on to form a stem canker like that. We get a partial canker, and that's what we term as our adult plant resistance. So over time, when we talk more about resistance, that's what we're talking about. So if anyone says, we only have adult plant resistance, it's probably not true. You probably do have seedling resistance as well. It's just that it's been overcome. And there are cultivars that do have this effect. Okay, so that's the resistance story. Just moving on to the fungicides. This slide here is just looking at infection rates. There's our canker percentage, and there's plant death. Compared to when we actually inoculate the plants. So we grew plants up we sow them at different times, and then we put them all out in the field on the same day. So the plants range from the first leaf to the rosette stage. We looked at how much disease they get. And you can see here by the 5th leaf stage, we no longer get any plant death, and the levels of stem canker have reduced greatly. So what we see is that what we need to do is really protect those seedlings. It's the seedling infection which causes the stem canker, and the canker infection you see at the end of the year is always a result of that seedling infection. So basically, the plants which are infected later in the year basically escape the disease. The A is not too far to maturity, and B, the fungus has got a long way to grow if it's landing on the 7th leaf to get down to the crown. So as a visual representative, this is what you want to avoid. When you see plants colonised like this at the seedling stage, and you can see the petioles already dying, then the pathogen's only got to grow about 1 centimetre to get down to the crown. That's severe levels of disease, and you can see here this is just a seedling growing. This is canola on canola. You can see the stubble there with the fruiting bodies on it. And so really with all our fungicide strategies, it's these guys here that we're trying to protect, and that'll give us less stem canker at the end of the year. This is an older slide that Trent Potter put together a few years ago, and it looks at the effect of jockey and impact. But I think it sums up the story quite well. There's a lot of work done at jockey at the time. So this is yield. It's canola on canola, so it's obviously very high disease pressure, done at Marcroft. And we've got a range of cultivars from highly resistant, in fact at the time that was immune, down to ones with only moderate levels of resistance. And you can see here, even under that very high levels of disease, if you're growing a good resistant cultivar, the fungicides, the jockey, the impact, and the combination don't give you much benefit at all. If you're growing something with low levels of resistance, you can double your yield, but it doesn't actually bring you back up to yield potential. So they're giving you a boost, but they're certainly not completely controlling the disease. But we're getting our best results when you've got that moderate to a good level of resistance, and then the fungicides are giving you a really good economic pickup. Some work that John Kierkegaard did from CSIRO when he just went through, basically trawled through all the literature that had been done on the two and looked at the yield responses. So in 26 out of 89 cases where there was a yield response, the average yield response with jockey was 0.1 of a tonne. With impact, it was considerably better at 0.17. Interestingly, when I was talking to the guys in the Lower Eyre Peninsula, they've got the same data and their average yield response with jockey is over 20%. So it just shows for the fungicides, if you're in really extreme situations, you get a massive pickup with the fungicides, but for a lot of other people, you're not going to. And that's, again, why it's crucial to know how much disease is actually in your own cultivars. John also looked at comparing jockey with impact, and in 42 trials where they were both in the same trials, 1 out of 42 the jockey was better than impact, 17 out of 42 they were equal, and 24 out of 42 the impact was better than the jockey. Now, moving along to foliar fungicides. So before Prosaro was released, we did some work in the disease nursery. We didn't have Prosaro, so we used impact and furrow as a foliar. And we just looked at the timing of application, because it said we'd done that work which showed you had to really protect that seedling. So this is plant survival, canola on canola, and you can see with no fungicide, you have virtually no plant survival. You nearly have 100% plant death with the cultivar cobbler. And then we started putting our fungicide on at different growth stages, and we started, getting some good significant increases in plant survival. The interesting thing here is here's jockey by itself, and it is considerably better than any of the foliar application by itself. So if you're going to have a choice about using a fungicide, jockey is certainly the preference. But then when we use jockey in combination with the foliar fungicide, we start getting a pickup again. And you can see here that the jockey and then the 6-leaf application or 6 and 4-leaf applications were the best. So you're using your jockey to protect the seedlings that comes out of the ground, and then you're using your foliar to try and extend the level of that protection for a little bit longer. This is the same work done now with Prosaro. This was at an NVT site, so it was just normal canola production, 2 cultivars which had various levels of blackleg infection. Here's our control with just over 50% internal infection when we cut them open. And again, here's our Prosaros. So we've got very good control of the Prosaros. But again, here's our jockey. So our jockey is still better than the Prosaros. And then the combination of the jockey and the Prosaro are certainly the best, and we've almost got complete control there. And that's now the industry recommendation of jockey plus the 4-leaf. So the main point with this work has been that there's no point using Prosaro unless you've already protected your seed with an impact or a jockey beforehand. You've got to protect that seedling as it comes out of the ground, and then use your foliar to extend the life of that protection. This is where it gets interesting for you guys in making recommendations. So this is exactly the same trial with the same cultivars, and here's the yield responses to all those fungicides, and we got no significant yield response at all. So if you go back to this slide, if you remember at the beginning of the talk, I said you need to get around 50% internal infection to start getting the yield loss, and although we had reasonable levels of disease, it really wasn't quite high enough to start causing major yield loss. And this was done in 2011 when there was quite good subsoil moisture. So we also see with blackleg in those tight springs, a smaller infection will cause more yield loss. So with things like Prosaro, obviously if you're growing canola in the wrong situation, right next to last year's stubble with a cultivar with lower resistance or something like that, there's a very good chance of getting an economic yield response. But if you're not in those situations, you're probably not going to get a yield response. But again, it goes back to each year, monitor how much disease you've got, and then you can make an informed decision on whether you're going to spend the money or not. Next part of the talk is when we're going to move on to the resistance groups, which were released for the first time this year. So the concept here is that, I said at the beginning of the talk, you control the pathogen by which cultivar you grow. So if you grow a particular cultivar year after year, you will select isolates which can attack that cultivar, and you'll end up with a particular population which can attack that cultivar. But by switching cultivars to one which has a different resistance mechanism, you can overcome a lot of the disease severity by doing that. So this is just field observations of what occurred on Eyre Peninsula after 2002 when we had the sylvestris resistance. So in 2002, all the farmers were growing sylvestris resistance. It was a seedling major gene resistance. It was immune to the disease, and we got no blackleg infection. Now in 2003, that resistance gene was overcome by the pathogen. The crops no longer had any resistance at all, and they all died. By 2005, obviously after 2003, farmers didn't grow it at all. 2005, those sylvestris plants started coming alive again because the infection levels from that stubble had dropped dramatically. At the same time, here's what we call our non-sylvestris resistance. In the same trials, here's a cultivar, it was Beacon. It used to get lots of disease, well, a moderate level of disease. When the populations totally switched to kill sylvestris, they actually lost their ability to attack Beacon. So the Beacons actually then became immune. Then everyone switched from this resistance back to the Beacon, and the sylvestris started coming alive again, and lo and behold, we saw the Beacon starting to get disease. So from these field observations, we saw quite clearly that we can control what the blackleg populations are in the field by determining which cultivars we grow. So we came up with the idea that we really need, when we're growing stuff like this, we need to switch to this before that happens. So we basically spent the last 10 years trying to devise a plan for being able to do that, and like I said, we released it this year. So the first thing we need to do is work out what resistance is in each cultivar. So there's no point switching cultivars if they've got the same resistance genes. So we have a system of 14 different blackleg isolates which can attack known resistance genes, and we use them and we look to see if they can attack or can't attack, and from that we can identify the seedling and resistance genes in each cultivar. And as I said before, all our cultivars have seedling resistance, although they're not always effective in the field. The important thing then is to work out the adult plant resistance as well. Now, adult plant resistance is a really tough one to crack. It can be done, but it's incredibly laborious. So we've developed this system where we classify all the cultivars on their seedling resistance, and we put them in 7 different groups. We then take stubble from each of those 7 different groups, and we allow that stubble to infect every individual cultivar, so 7 times. And this is the sort of effects we get. So this is whole of 50, which is R-rated. It's very, very, very good. But when we attack it with its own, so it's a Group D cultivar, when we attack it with its own stubble, we kill it. When we take exactly the same cultivar but attack it with a Group B stubble, it survives. We do it the other way around. Here's our Group B with our Group B stubble, we kill it. And here's our Group B with that same Group D stubble, and it survives. So basically, this is the process. We use the seedling resistance and then this stubble screen to classify all our cultivars now into 7 different rotational groups. Now, that's all in the blackleg management guide. So you can see here we just have a step 4 blackleg resistance groups and a bit of methodology. So you can see you've got your cultivar here, whole of 50, it's R-rated, and then it's blackleg rating with the jockey as well, and then which resistance group it's in. And you can see here, just to complicate things, it's R-rated, which means it should have extremely high resistance, but put it in the wrong situation, it can become totally susceptible. So that's what you need to keep up with now. The other thing we're doing with this system is we're trying to provide monitoring data across Australia. So we're monitoring the 7 different populations and trying to provide the information back to you guys. So we grow 1 cultivar from each of the 7 different groups, and then we go around and we cut them all open and look to see how much internal infection there is. We've got, said, 32 sites across Australia co-located with MVT trials. So from now on, when you go onto the MVT online website and look at the yield data for a region, you'll also get the data from these blackleg monitoring sites. And this is the type of data we're generating for each site. So these are some sites from 2012. You see here this site in New South Wales. We had high levels. Sorry, this data is internal infection data for that site compared to the national average. And you can see here on that site we've got a problem with Group A, but all the other groups are all fine. This is the one from the Eyre Peninsula where we grew lots of whole of 50 and we've now destroyed whole of 50 on the Eyre Peninsula, but all the other groups are standing up quite nicely. We've got a group there where we're getting increasing populations in 2 of the groups, and obviously there's an example of one where there's no particular issues at all. One of the things is this data needs a fair bit of interpretation, so we've decided not to put it out like that. We've decided to interpret the data for you, and this will be what you see when you go onto the NVT website. You'll get basically a paragraph saying which groups have been overcome or not, and then some recommendations for actually using the groups and using the system. One of the key points to remember is all we can monitor is what the population is at that individual NVT trial, which is why we still say it's very important to have some idea of what cultivars you're growing and how much disease they have on your own farm. From that data, we can also put out warnings, and they will happen from time to time when we think there's a major issue. This is the warning we put out 12 months ago when our monitoring site showed the whole of 50 was coming unstuck. It had gone from basically being 0 internal infection to 70% internal infection over 3 years. So we put out the warning not to grow it anymore. And because we had our data, we could also say which other cultivars had the same resistance and put out a warning for those cultivars as well. And that actually resulted in no Group D cultivars being grown on Eyre Peninsula last year. And when we went back to our monitoring trials last year to see what actually happened, this is what we saw. So here's our Group D, whole of 50, which, if you remember, was immune to the disease. It was R-rated, and this is what it looked like last year. Here's our Group E, and which in exactly the same trial, virtually had no disease on it at all. These were both R-rated, so we couldn't tell the difference. They're from the same seed company, but our monitoring system of the resistance genes said no, they are different, and that was verified in the field last year. And the really exciting thing is that when the Sylvestris breakdown happened, we told the farmers no longer to grow it after their crops looked like this. It was a little bit late, whereas in this situation, we managed to do it the year before this occurred. So it was a fantastic result for the industry. The other thing with all our monitoring trials was that we only saw the problem on the Eyre Peninsula, so that we could only put out a regional-based warning just for the Eyre Peninsula, and everywhere else around Australia continued to grow those Group D cultivars. And again, when we came back this year, that proved to be true. So when the Sylvestris broke down in 2003, we basically said no one in Australia should grow Sylvestris anymore, whereas this time we could be actually regionally specific and say, yes, it's occurred on Eyre Peninsula, but there's no evidence of any problems anywhere else. And with our monitoring sites each year, we will then be able to tell people when not to pull out of these type of groups and switch. One of the things with all these warnings too is that in the past we used to put loss of resistance warnings on the blackleg ratings. We've decided not to do that anymore. We're really going to switch back to this system where we'll put out specific warnings for regions, and hopefully you can also look at the data and know, make basically much more on-farm decisions about. So if you've grown the same resistance group for 3 or 4 years, you've seen disease increasing in it, you will then switch to a different resistance group and manage that with your own knowledge on your own farm. And hopefully we'll provide the tools for you to do that. So that's the end of the blackleg talk. So we're just moving now on to Sclerotinia, and Kurt Limbeck from New South Wales put this talk together. So last year there was quite a bit of Sclerotinia in New South Wales, and obviously northeastern Victoria as well. And just running through again why Sclerotinia is also a very difficult disease to crack. This is the life cycle for Sclerotinia. So it starts its life as a sclerotia, which looks like a little black rat turd, and it survives in the soil, can survive for 8-10 years in the soil. When its conditions are right, it germinates, produces a little mushroom which produces spores. The spores are wind-blown, they land on the petals, and they just stay on the petals. When the petals fall off the plant and they get stuck in a branch, something like that, that's when the spores germinate and they start colonising and using the petal as a food source, and then they grow directly into the stem. So they can't grow directly into the stem without going through that petal stage as well. And then they produce, inside the stem, they produce more sclerotia, which go back into the soil. One of the tricks with Sclerotinia, though, is that it's an incredibly sporadic disease. It has to have the right weather conditions all the time for it to actually occur. So Kurt's put these clouds in it. You need warm, moist conditions at those growth stages for you then to see this level of infection. So if you miss rainfall in any of those stages, it doesn't occur. So, which is why it's very hard to say you're going to get a Sclerotinia problem this year and not next year or whatever. That right out of the blue, you can get a problem if that all lines up. On the flip side, you can grow really good crops and have no Sclerotinia problems at all. So it's very hard to model whether you're going to get a problem or not. So sporadic disease, and it also varies enormously on flowering time. When the crops are flowering, depending on when the rainfall occurs. So one year you'll get the early sown crops will get Sclerotinia, the next year it'll be the late sown crop. 1 year in the MVT trials, the early flowering ones will get more Sclerotinia than the late, and then the next year it'll be switched around again. So it's very hard to pick. It also, from a control perspective, it's hosted by around 400 species. So all the pulses, all the broadleaf weeds and everything can get and harbour Sclerotinia. So rotation is incredibly difficult. And again, it's very hard to predict because of the seasonal conditions. Kirst has put in some photos here of Sclerotinia. There's a badly affected crop from last year in New South Wales. Here's the typical symptoms where the infection started at that point there and it's grown up and down and it's really bleached white and very hard. And when you crack it open, you see these sclerotia, which will survive for many years. Another photo of bad infection in a crop. It's a photo of where Kurtz has had an infected plant there, cracked it open, and you can see the sclerotia. Sometimes you'll get the sclerotia forming on the outside, but it's much more typical to see them on the inside. So if you're ever wondering what it is, if you're cracking them open and you see that, it's definitely Sclerotinia. Now, this slide here is some observations that Kurt made in New South Wales on primary wheat canola rotations. And what you're looking at here is still the Sclerotinia infection, but this is the root down here. So the infection, Kurt believes, has actually come directly through the soil. So where you've had wheat canola, there's been a build-up of sclerotia to a high level in the soil. And then when this year's crop has been sown right basically on top of sclerotia from last year, and those sclerotia have germinated in the soil and they've actually directly infected this year's crop. So if 2012 was a bad Sclerotinia problem, if you put canola crops back in 2014 and the conditions are right, we might see a lot more of this basal type infection. And so this is a lot of anecdotal information at the moment. Kurt really needs to do some more work, but it's a bit of a worry. And he saw it up to about 30% in some crops, but typically around 10% across New South Wales of the infections were actually at the ground level, this basal type of infection. And the trouble with this is that the foliar fungicides aren't going to have any control over this type of Sclerotinia. So Kurt's observations from last year, he saw infection levels of up to 70%, and as a rule of thumb, if you halve that level, that'll give you an idea of what the yield loss will be. So we're in some individual situations seeing some very high levels of yield loss. Unfortunately, these wheat rotations appear to be the worst affected with this basal stem of infection just from the level of sclerotes, which is going to be something which we may have to deal with in the future. Now, spraying. Kurt's done quite a few trials over a number of years on spraying for control, and it reigns true with what they find in Canada and Europe, is that you really do need to spray that 20 to 30% bloom. And this photo here is of a crop at 30% flowering, and that's an individual plant, 30% flowering. Kurt said a lot of farmers are trying to put it off a little bit later to try and protect more petals, but he said the really important thing is that you get your chemistry through the canopy down to actually get some chemical on your stems where the petals are going to fall. So it's really crucial to actually spray at this time and not to leave it to a little bit later. It's very, very difficult though to make economic decisions on when or when not to spray, because you never know if you're going to get the disease or not, and it's reasonably expensive. So his rule of thumb is if you're growing if your crops have got really good yield potential and you're getting that wet spring weather during flowering period, that's a good indicator to go out and spray. And Kurt just makes the other point about which products are registered. So that's where I'll leave it. Any questions?
Okay, thanks, Steve. There's one texted question up there. We've probably got time for one from the floor.
2 years ago, for want of a better term, we saw infection cankers forming high up in the plant. We thought it might have been, there's 2 species of blackleg, and we thought it might have been the other species because that's what it does in Europe. But we did test it and it's not. It's the same species that we currently have in Australia. We've just termed it, for want of a better term, canker in the branches. We only saw it, that was 2010. We only saw it in one region in Victoria, but Kurt picked it up in a few locations in New South Wales last year. So yes, it can. And that's about all we know about it at this stage. We really haven't done any work on it. We've seen it twice now in the last 3 years. And whether it's going to become an issue for us or not, I don't know. And then unfortunately, this was in a cultivar which was very highly blackleg rated and had no stem canker.